I welcome you at Advanced Multiple YouTube channel. Advanced Multiple Incorporation actually is knowledge processing and information management organization that is located in Mississauga, Ontario, Canada. My name is Dr. Taha Nazir and working as the scientific executive with Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Both of these organizations located in Mississauga, Ontario, Canada. I'm going to talk about my research under the title Practical Pharmacokinetic for Best Pharmaceutical Care in Different Hospitals or Clinical Setting. For this purpose, I collected the information from different scientific data sources like PubMed, Medline, Scopus, Google Scholar, National Library of Medicine, MBAS, Directory of Open Access Journal, ProQuest, CNKI, that is China-based database company that provides the scientific research information all over the world. Some of information collected from ResearchGate and Crossweb. Okay, well, before moving forward, I would like to discuss a little bit about the practical pharmacokinetic. Practical pharmacokinetic or therapeutic drug monitoring (TDM) is a branch of clinical chemistry or clinical pharmacology that deal in the measurement of drug concentration in the blood of patient. Therapeutic drug monitoring used to detect poisoning or toxicity induced by potent control or narcotic drugs in clinical setting or in hospitals. Now, I would like to talk a little bit about TDM or therapeutic range. Main focus of clinical pharmacokinetic is to closely monitor the drug with the narrow therapeutic range or drug that can easily become under or overdose. For example, aminoglycosides comprise of drug gentamicin, kanamycin, streptomycin, then anti-epileptic drug carbamazepine, phenytoin, and valproic acid, mood stabilizer, lithium citrate, and antipsychotic drug pimozide and clozapine. These drugs have very close or narrow therapeutic range, and they become toxic right away, or they may remain underdosed. So we have to monitor its concentration inside the body of patient to maintain the therapeutic range or keep the concentration of the drug in between the therapeutic range to obtain maximum pharmacological effect. This is the diagram actually indicating different parameters used in therapeutic drug monitoring. It is very simple linear graph. When we say linear graph, it means there should be mostly two parameters used in linear graph. In horizontal, we have time in hours. In vertical parameter, in vertical section, we have plasma drug concentration, unit milligram per ml. In this diagram, we have different points. Most important points are four. The first one is minimum toxic concentration. This is the minimum concentration of the drug that may potentially produce the toxicity in a defined particular population. Then second point is minimum effective concentration, that is the minimum quantity of the drug that may produce therapeutic effect. Uh, Below this concentration, it may be useless. It may not produce pharmacological desired effect. And if it is more than minimum toxic concentration, that it may be toxic. It may produce adverse effect in the body of the patient. So we have to keep the drug inside the patient under therapeutic range. Therapeutic range is between the minimum toxic concentration and minimum effective concentration that we call as steady state. This steady state concentration is more desired concentration to obtain the maximum pharmacological effect in clinical setting or in hospitals. Now we are going to talk a little bit about the scope of TDM. TDM used to improve patient safety and care by individualizing or adjustment of the dose of drug for each patient. It also used for scientific experiments in the research laboratories or clinical trial to improve the therapeutic benefit in general of particular population. TDM benefit based on pharmacogenetic, demographic, pharmacokinetic, or pharmacodynamic or clinical information of patient. When we say clinical information are four main types. It may be clinical, it may be physical, it may be personal or professional. The clinical information, we know it may be the liver function test, maybe renal function test, may relate to the blood chemistry, or it may relate it to the respiratory function. It actually related to the physiology and pharmacology of the patient. Then physical factors or the physical information, body weight, gender, and height. Then personal information mean name, address, location. Professional mean what kind of the professional skills or expertise or occupation patient have. So we collect information before and after administration of the drug for the measurement of accurate drug concentration, pharmacokinetic parameters, biological impulses, endpoint markers of effect are collected through pharmacodynamic monitoring. So the overall data used to improve the clinical and pharmaceutical patient care for, hospitalized patients. Now, I would like to discuss a little bit about workflow of therapeutic drug monitoring or the functionality or the operation of therapeutic drug monitoring. There are the three main categories of therapeutic drug monitoring: category A, B, and C. Category A was introduced approximately 50 years before, that was then replaced with this B, and nowadays, most current technology or most current uh, like techniques used in therapeutic drug monitoring, they fall in category C. We see that there are the five steps involved in category A. They are then converted into the two, and finally, nowadays, there are the two steps involved in therapeutic drug monitoring. In category A, we see that that is called as traditional TDM, patient transfer from the home to the hospital, to the transplant center, to the clinics, from where if it's different samples are collected. If you say that sample, that may be the biopsy fluid, maybe the urine, maybe the blood, or maybe the bronchial washing or sputum. These biological samples are then prepared and sent to the research or clinical lab or the diagnostic lab. In diagnostic lab different procedure techniques and equipments are used to analyze the sample and obtain the information. These diagnostic information that's sent to the uh, clinic or hospital again, where expert review the information. Expert mean it may be the clinician, pharmacist, physician, staff nurses. They work together like a team and then design more rational treatment protocol. More rational treatment protocol mean they decide the dose, regime, protocol for the treatment of particular diseases for potent drugs. In category B, we have three steps. The facility center, where the different samples collected. Samples as we have discussed before, it may be the biopsy fluid, maybe the urine, maybe some like sputum. It is then sent to the diagnostic center. Information regarding the diagnosis sent to the like clinician or the physicians. They analyze, work together, discuss, and design the more rational treatment protocol. In category C, there are only two steps involved: the patient side, that is the clinic or hospital, and the transplant center, where the uh, biological samples are analyzed, investigated, and the most rational treatment is designed. There are the different variables that influence the drug concentration. There are various variables that influence the interpretation of drug concentration. Drug related variables are time, route, dose, time of the blood sampling, handling, storage condition, precision and accuracy of analytical method, validity of pharmacokinetic models, and co-medication. The patient-related variables are clinical status of patients, disease, renal, hepatic status, biological tolerance to the drug therapy. Miscellaneous factors are method, equipment, protocols, techniques, and environment and professional expertise that may influence or affect the drug concentration in patient. There are the multiple people, multiple roles involved in TDM. Many different professionals are involved in TDM are physician, clinician, pharmacologist, clinical pharmacists, staff nurses, medical laboratory technicians, scientists, statistician, and bioinformatics. These all people work together to perform a single role that is called as TDM. Various elements used to monitor the drug concentration and take decision regarding the treatment of patient. Failure to carry out any one of the component can reduce usefulness and pose potential challenges for the physicians. Drug concentration monitor help to optimize therapy and organize the approach in clinical or more critical manner. Characteristic of therapeutic drug monitoring in pharmacotherapy: many medications are monitored in blood because their dosage can generally be varied in clinical response. In small group of drugs, insufficient level will lead to under treatment or resistant or excessive level of the drug that may lead to the toxicity. Element of therapeutic drug monitoring are determination of relationship between the plasma drug concentration and pharmacological effect. Knowledge of the drug level influencing patient physiology, therapeutic window, patient compliance problem, drug dose, and clinical observation of the effects. In conclusion, TDM can potentially be used for rationalization of the treatment in clinical setting. TDM involves the determination of various factors related to drug, patient, and environment. These scientific factors consider to achieve the desired medical targets. The goal of TDM is to use appropriate concentration of difficult to manage medication to optimize clinical outcome in patients having challenging clinical situation. I would like to salutate and greet to my colleague, my team, my coworkers, Sherry, Val, Daniel, Dustin Hawker, Marwa, Robert, Hunza, Noreen, Terry, Istiaq, Arsanan, and Amin. I also acknowledge the kind support and help of Advanced Multiple Incorporation and the Intellectual Consortium of Drug Discovery and Technology Development Incorporation. I appreciate the support and help these two organizations. Uh, Thank you very much for staying with me and watching the video. I hope we will meet in our next video. Thanks again. Take care. Stay blessed. See you soon. Bye now.